Hello viewers, welcome to the series of lecture in food technology. Today I am going to deliver the lecture on the topic pectins. The word pectin derived from Greek, it means congealed. It has been recognized for at least 200 years and was originally identified in 1790 in apples by the French chemist Nicolas Vauquelin, who also discovered the elements chromium and beryllium. Until 1984, there were no further reports available on pectins, and later the scientist Braconnet was undertaken. He has named the acid gelling substance pectic acid after Greek word for gelling or congealing. Further, Smolensky identified the gelling substance as a polymer of glucouronic acid, and later on in 1937 Schindler and Bock established basic formula of pectin. Pectins are the mixture of polysaccharides which is found as a major component of cell walls in all plants, particularly apples or citrus fruits. Pectins are complex heteropolysaccharide composed primarily of essentially linear polymers of alpha D galactopyranosyl uronic acid units linked through alpha to delta or 1 to 4th glycosidic linkages. The polymer chains are esterified to various degree with methanol. This regular structure is interrupted, however, with L-rhamnopyranosyl units and with side chains containing other neutral sugars. The polymer chains may also be partially acetylated. The most important physical property of pectin is its ability to form spreadable gels. Gel formation results when the polymer chains interact over a portion of their length to form a three-dimensional network. This aggregation of chains occurs through hydrogen bonding, divalent cation cross bridging or hydrophobic interactions. Pectin contains pectinic acids as a major components, water soluble and able to form gels under appropriate conditions. In this episode, the following 5 divisions have been studied on pectins. 1. Sources of pectins. 2. Properties of pectins. 3. Classification of pectins. 4. Extraction of pectins. And finally, application of pectins. Let us see sources of pectins. Presently, traditional commercial sources of pectin have been employed extracted from citrus peel and apple pomaceae, which are byproduct of juice manufacturing units. Citrus peel, generally lemon and lime peel, are one of the most preferred sources for the manufacture of pectin due to its high pectin content and good coloring properties. Pectin can convert to pectic acid upon the hydrolysis of lime treated peel and further treated with enzyme to ease the peel removal. Apple pomaceae contains 10 to 15% of pectins, whereas citrus peel contains relatively higher, that is 20 to 30% of pectins. More recently, other sources of pectins are beginning to search for market, including sugar beet waste from sugar manufacturing, sunflower heads seed used for edible oil, and mango waste. Also, crab apples, gooseberries, some type of plums, I bush cranberries, strawberries, cherries, blueberries and papaya contains little percentage of pectins. Next, properties of pectins. The gelation properties of pectins can be divided into two main categories: high methoxygelation and low methoxygelation. High methoxygelation pectin usually forms at a pH of below 3.5 and total solids content of above 55%. This type of gel formed during jam making, and high methoxypectins are characterized by their setting time and gel strength. Setting time is usually categorized as rapid set, medium set and slow set. High methoxypectins gel slower as more of the methoxy groups are removed during processing. Low methoxypectin is gelled with calcium ions and hence is not dependent on the presence of acid or high solids content. The low methoxypectin has the lowest level of esterification, and amidation can interfere with the gelation, causing the gelation to be delayed. Another useful property of amidated pectin is the ability of the gel to heal after sharing. Monovalent cation salts of pectinic and pectic acids are usually soluble in water. Di and trivalent cation salts are weakly soluble or insoluble. Dry powdered pectin, when added to water, has a tendency to hydrate very rapidly, forming clumps. These clumps consist of semi-dry packets of pectin contained in an envelope of highly hydrated outer coating. Clump formation can be prevented by dry mixing pectin powder with water-soluble carrier material or by the use of pectin having improved dispersibility through special treatment during manufacturing. The composition and chemical structure of elements that constitute pectin depends on environmental conditions and plant sources. In addition, pectin structure is modified by enzymatic and chemical reactions during plant growth. Fruit ripening and extraction techniques are conditions. All of the above factors make the structure and molecular weight of pectin to vary from plants to plants and even among tissues in the same plants. Therefore, chemical structure and composition of pectins are studied by classifying them into native and commercial pectins. Native pectins is composed of three major polysaccharides, all containing alpha 1 to 4 D galactouronic acid residues, usually referred as galactouronans. There are homogalactouronans, rhamnogalactouronan 1 and rhamnogalactouronan 2. Homogalactouronan is a linear chain of alpha 1 to 4 D galactouronic acid residue with a variable degree of methyl esterification at the carboxyl group. It could be O-acetylated at C2 or C3 depending on the source. Rhamnogalactouronan 1 consists of repeating units of the disaccharide alpha 1 to L-rhamnose alpha 1,4 D galactouronic acid. Galactouronic acid residues can be O-acetylated at C2 or C3, while 20 to 80% of rhamnose residues can be substituted at C4 or C3 with neutral sugar side chains. The composition of the neutral sugars varies among plant sources as D-galactose, L-arabinose and D-xylose being the most common. Other neutral sugar such as D-glucose, D-mannose, L-fucose and D-glucuronic acid are found less frequently. Rhamnogalactouranon 2 represents its misleading identification as a backbone of alpha-1,4-D-galactouronic acid. Some of the side chains attached to the backbone have been identified as 2-keto-3-deoxy-D-mannoctolysinic acid, 3-deoxy-T-lyxo-2-eptolysoric acid, epiose and azeric acid. All neutral sugars are located inside chains or rhamnogalactouronons 1 and 2 domains, and therefore these domains are often referred as hairy regions. In addition to the above three major domains, arabinogalactons, arabinons and xylolactouronons are also found in native pectin, all lacking the galactouronon backbone. Commercial pectins are structurally less complex due to industrial extraction and purification process, which remove most of the neutral sugars. This consists mainly a backbone of alpha-1,4-D-galactouronic acid with partial methyl esterification of the carboxyl groups. At least 65% of extracted material must be glucuronic acid in order for the extraction material to be classified as commercial pectin. Pectins are subdivided according to their degree of esterification. A designation of percent of carboxylic group esterified with methanol pectins with degree of esterification greater than 50% are highly methoxy pectins. Those with degree of esterification less than 50% are low methoxy pectins. The degree of amidation indicates the percent of carboxyl group in the amide form. Rapid set of pectin is a high methoxy form of the substance, meaning it contains high quantities of compounds methoxy group. It is the most often used in preserves that contain bits of fruits or vegetable, such as jams and marmalades. Slow set pectins, another high methoxy form of pectin, is used in less pulp laden jellies such as apricot and grape jelly. It is also used in some less pulpy jams and preserves. Stabilizing pectins are most commonly used in dairy products. Specifically, they are used to stabilize acidic proteins in dairy products during any heat processing used to manufacture the products. Stabilizing pectins can be found in yogurt, soya beverages and whey beverages. Low methoxy pectin, also known as LM pectin, is often used for food products with reduced sugar content. This form of pectin needs calcium as a catalyst to begin the gelling process. Products containing LM pectin includes reduced sugar preserves, sauces, marmalades and dessert topping. LM pectin is also used in low acid fruit preserves such as fig preserves. Extraction of pectins. The processing of apple fruits for juice and cider making results in large amount of solid byproducts, which are termed apple pomace. Apple pomace approximately accounts 25 to 35% of fresh apple fruits that are processed. Apple pomace is mainly composed of carbohydrates, fibers, proteins and small quantities of minerals, and also considered a rich source of dietary fiber, especially pectin with 10 to 15% content. The feasibility of the processing operation of pectin production and its wide use in various applications make pectin extraction an effective way to utilize apple pomaceae. Pectin production is sometimes considered an art rather than science because a little vary in the experimental parameter such as temperature, pH, time, etc. will vary the pectin characteristics such as molecular weight, degree of esterification, yield, etc. Pectin extraction is a multi-stage physicochemical process where pectin molecules are hydrolyzed and solubilized from the cell wall and middle lamella of the plant tissue. Extraction conditions used in the process vary depending upon the raw material, type of pectin and process economics. Commercial pectin is usually produced by hot acid extraction followed by filtration, alcohol precipitation, drying, grinding and standardization. Depending on the chemical characteristics of pectin, it is used in numerous food applications as a gelling agent, thickener, stabilizer and emulsifier. The traditional application of pectin is a gelling agent in a wide range of fruit-based products like jam, marmalades, jellies, fruit precipitations for yogurts and desserts, fruit filling for bakery products. It can be used to improve the mouth feel and the pulp stability in juice-based drinks and as a stabilizer in acidic protein beverages, and also reduces the synersis in jams and marmalades and also increase the gel strength of low calories jams. Pectin has a potential to be used in the encapsulation of unstable food ingredients such as high methylated pectins are used in jams and jellies. The role of the pectin is to impart a gel texture to the jam or jelly. This texture must be strong enough to allow transportation and storage without changes. It is mainly used for fruit flavored products where the acidity enhances the most fruit flavors. The pectin is used within the confectionery industries for making fruit jellies, jelly centers and wine gums. Stabilization of protein containing drinks with pH around 4 is an increasingly an important application area for pectin. Pectin is also used for fat replacement in low fat spreads and butter spreads. In these products, it binds water and thereby improves emulsion stability. A pectin type splendid has been developed to mimic a fat-like mouth feel. Pectin is used in fruit milk desserts where the interaction within calcium is used to obtain an instant gelation when adding calcium ion to syrup containing pectin. Alpha tocopherol, which is the most abundant and active form of vitamin E, is commonly used by the food industry for food fortification and to inhibit lipid oxidation. However, the use of alpha tocopherol is sometimes hindered by its sensitivity to heat, oxygen and light and also high hydrophobicity. So, pectin could be used as an ingredient in the development of polymeric matrix for the encapsulation of alpha tocopherol to provide protection, enhance the stability and also to deliver alpha tocopherol under specific conditions or gastrointestinal environments. Pectins are also used in pharmaceutical and also in cosmetics. Pectins has multiple beneficial effects on human health, including lowering of blood cholesterol and serum glucose levels and potential inhibition of cancer growth and metastasis. Some of these benefits may occur via the induction of apoptosis and also interfering with ligand receptor interactions. Irritable bowel syndrome, a functional gastrointestinal disorder, can be reduced by fiber diet means through pectin. In fact, one of the leading medications for this problem, Pectin, used pectin as one of its main active ingredients. Here pectin acts as thickening agent and also found in other medicines such as Levaden, it is a cough syrups. Orange peel pectin shows a good potency as a binding agent. The properties of pectin are utilizing in a variety of personal care applications, including skin and hair care products and also color cosmetics. Apart from providing structure in formulated products, pectin can form a gel on the skin. The viscosity of pectin solution is utilized to evenly distribute and provide non-slimy spreadability of air conditioner and hair styling products. In conclusion, pectin is a polysaccharides which are localized in the primary cell wall and middle lamella in all higher plants. Pectins are responsible for different physiological processes like as one of the main agent cementing the cellulose fibrils and may be linked covalently to other polymers. Commercial pectins are extracted at low pH and high temperature. Pectins are widely used as food additives with gelling and stabilizing properties in jams, jellies, milks and confectionery products. Pectins can be categorized according to their degree of esterification, a designation of the percent of carboxyl group esterified with methanol. Pectins with degree of esterification greater than 50% are higher methoxypectins. Those with the degree of esterification less than 50% are low methoxypectins. The extraction of pectin is a multi-stage physicochemical process by hydrolyzed and solubilized from the cell wall and middle lamella of the plant tissue. The conditions used for the extraction process varies depending upon the raw material, desired type of pectin and process economics. Thank you.